Okay, we might get started, everyone. Thank you so much for joining us this evening for our July Geography Matters. Tonight, we'll be hearing from Dr. Mary Droser and Associate Professor Diego Garcia Bellido, who'll be discussing the significance of the Ediacaran fossil record of South Australia for our understanding of the unfolding of animal life on Earth. Before we get started, a little bit of quick housekeeping. As always with our Geography Matters webinars, we are recording tonight. So please do make sure that you keep your microphone and camera off so that you're not added into the recording. We will, of course, have plenty of time for questions at the end this evening. So if and as something comes to mind, please do throw it into the meeting chat, and our wonderful attendees or presenters this evening will be more than willing to answer those questions and dive into them in more detail. Additionally, if you do have any problems throughout the evening, do message myself. More than happy to assist as needed. So, as we get started, I would like to acknowledge the traditional custodians of the lands on which we meet and recognise their continuing connection to land, waters and culture. For myself this evening, that's the Yagara and Turrbal peoples here in Brisbane, and I know we've got plenty from across Australia joining us as well tonight. A quick plug, as always, the RGSQ has a vibrant program of events and activities that are constantly rolling throughout the year. For those who are already on our mailing lists, you will have seen one earlier today with what's coming up in the next month. For those who aren't already, please do check out the RGSQ website for more details about what we have lined up over the coming weeks. Of particular highlight, though, is our next Geography and Conversation panel session coming in on July 23rd. We'll be looking at artificial intelligence in geography. We have our professionals talk series on the Thursday of that week, looking at careers in transport. And this time next month, we have a Geography Matters webinar looking at climate change in the Torres Strait. That's the 13th of August. So please do keep an eye out for those and register as soon as you can while there's still places available. Without further ado, though, I'll introduce our wonderful speakers and we can dive into the presentation. Tonight, we have Mary Droser, who's a distinguished professor at the University of California, Riverside. She works in the area of evolutionary paleoecology, examining large-scale interactions between life and environments. In the last 20 years with Australian colleagues, she's focused on the Ediacaran of South Australia, publishing over 50 journal articles on this extraordinary fossil record. Topics include the earliest sexual reproduction, descriptions of new body plans, and the advent of mobility, and so on. She received the National Academy of Sciences Walcott Medal and the 2024 Paleontological Society Medal for her work on the Ediacara Biota. Her interviewer this evening is Dr. Diego Garcia Bellido, who was born and raised in Spain, where he got his Bachelor of Science in Zoology and PhD in Paleontology from the Complutense University in Madrid. Almost got it. Diego has now settled in Australia, but has previously carried out long research stays in Canada, the US, UK, and Germany. He studies the early evolution of complex life during the Ediacaran and Cambrian 500 million years ago. He excavated at the famous Burgess Shale Cambrian fossil locality in Canada between 1995 and 2000 and worked at the Royal Ontario Museum in Toronto for several years. In 2007, he joined the Australian research team, excavating the Cambrian Emu Bay Shale in Kangaroo Island. And for the last decade, he's been studying the Ediacara Biota from the Flinders Ranges with the goal of finding the links of these iconic organisms and the Cambrian faunas. Without further ado, Mary, I'll throw to you.
Great, thanks very much. I will share my screen. Great. Okay, so I am going to start in the large scale, which is everybody on this Zoom, I think, has one time or another wondered, are we alone? With the discovery of so many new exoplanets, a new exoplanet a day, it feels like, we even are thinking about this even more than usual as we think about, is there habitability outside our solar system on some of these exoplanets or even some of the moons? But as of now, we have an N of 1, and that is there's only one planet that we know of that has life, that is covered with this green scum that we call life. And blue oceans and atmospheres, such as oxygenated atmospheres, such as we have on Earth. And so, you know, when we think about the history of the planet Earth, at one point it was just a hurtling rock going through space. We're still hurtling through space, but now it's covered with this life. And so one of the things we can do as we think about life on other planets is go back in time and look at the evolution of life on this planet, on our planet, and look at sort of alternative Earths. What was the Earth like 2 billion years ago, 1 billion years ago? And as you obviously know, I'm going to talk about a 500 million years ago. So 4.5 billion years ago, we had the origin of Earth. 3.5 billion years ago, it was a watery planet, and we have the beginnings of life on planet Earth, such as, oops, we see here. So the earliest record of life on planet Earth actually is also in Australia, in Western Australia, where we have 3.5 billion year old bacteria. And so we can look at this 4.5 billion years of Earth's history, which is such an unimaginable amount of time. And we have sort of bacteria-grade organisms up until about 1.5 billion years ago to 2 billion years ago. Then we get single-celled eukaryotes, organisms with DNA and a nucleus, which is a major breakthrough. And then about 500 million years ago, we get the advent of animals. And that's what I'm going to talk about today, is this early unfolding of animal life. How did animals actually unfold? What's the story of the unfolding of animal life on planet Earth? And so as you heard in the introduction, the dawn of animal life on planet Earth is represented by the Ediacara Biota. And this is just to give you the quick and dirty. It's a suite of fossilized soft-bodied organisms. They're called the Ediacara Biota after Ediacara, South Australia, which is about 20 kilometers from where I'm sitting right now. They are globally distributed, but there are a few important localities, and they span about 30 million years. And the Ediacara Biota in South Australia is not the oldest or the youngest, but within this 30 million years, it is by far the most diverse and includes many of the firsts that we think of in terms of animal evolution. It has the advent of true animals. And for those of you who are fans of the geologic timescale, the time period Ediacaran is named, it's the newest named time period in South Australia, the only one in the Southern Hemisphere. And years ago, actually, there was an Australian stamp series after the Ediacara Biota. So here's just a handful of shots of the Ediacara Biota, not our bearded dragon down here, but this fossil just below its nose. And you look at them and you're like, oh, these are really weird. I don't recognize any of them. They don't look like life as we know it. Maybe the upper left looks a little like something we can imagine. And so the first thing you think of or notice when you look at the Ediacara Biota is they look different than life today. And so one of our challenges and one of the things that we do working on the Ediacaran Biota, Ediacara Biota, is how do these fit in to our understanding of animal life on Earth? And so just a few more things that sort of set them aside from maybe what you might imagine. Sometimes people, when I say what I work on, they imagine they're really rare and maybe microscopic. When they occur, they're usually hugely common. They can be small on the order of millimeters or less, and they can be huge on the order of meters. Given that they're soft-bodied, if you think sort of sea pens, sea anemones, jellyfish, we don't necessarily think they are those organisms, but if you need a modern visual, they're still morphologically very diverse, and I think that will be a takeaway tonight. I hope so. And the other kind of crazy thing about these is probably everybody on this Zoom at one point either in your career or in 8th grade biology, learned about like kingdom, phylum, class, order, and how we classify living organisms. The Ediacara Biota, they are so strange and so different and different from each other that there are two interesting things. One, most genera are monospecific, not all, but most. That is, a genus only has one species. And they're not classified even into families, much like classes, orders, and so on, because we don't actually really know how they fit into sort of that classification of modern animals. But we're working on it. So just to put the Ediacara, Ediacara member, the work I'm going to talk about today, the locality I'm going to talk about today in context, and also, you know, sort of the global picture and temporal period. This is time going up here in 575 million years ago, the Cambrian up here. And so this is just time running up the left, and then range charts of different Ediacara taxa. And the one thing to notice from here, where you see the Ediacara member circled, is there are a lot more dots in that sort of medium gray bin. There was a much more significant number of different kinds of organisms during this time period that I'm going to talk about today. And it's the time where we see the advent of diverse and novel ways that animals behaved, the nature of their ecology, and their construction. So I'm going to talk about fossils at Nilpena Ediacara National Park, where I am currently seated. And I acknowledge that these lands where I'm seated and where this work was done lie within the Adnyamathanha traditional lands. And you can see in South Australia where I am currently seated, Nilpena Ediacara National Park. And this is a joint project between the South Australia Museum and the University of California. That's my colleague on the right there, Jim Gehling, with whom I have been working for now about over 25 years, and he's now retired from South Australia Museum. And for the last number of years, Diego and I have been working together, and here's just a picture of us with our joint group in the field. But this project has involved many, many people, South Australia Museum volunteers, many students, many family members. And this isn't even really a complete list, but so I'm going to be talking about this research, but it is based on the work of so many people, some physical, some intellectual, and so on. So just to give you a very quick idea of when I say the firsts in animal sort of first in animal evolution, some of the firsts that we record is the oldest record of active mobility, the oldest bilaterian, the oldest record of scavenging, oldest record of macroscopic life in the water column, and oldest record of sexual reproduction. And I'm going to, in multicellular organisms, I'm going to talk about a couple of these today, just to give you a flavor for what the record looks like. And so I'm not going to try and tell you everything there is to know about the Ediacara Biota. We don't either have enough time and you don't have enough patience. Most people, I don't have enough patience for that. But just to give you a flavor of what we're able to do and the nature of this early record of animal life. So first of all, what is the nature of the record? Why is South Australia special? Why is Nilpena now Nilpena Ediacara National Park? Well, first of all, the nature of this, we're in a shallow marine sea, so we're in an ocean. We are in relatively shallow water, but not water where you're going to go surfing, but sort of water that would be protected in a bay or something like that. That's our geography of this area at the time. And you can imagine a scene as you see above. Organisms are happy on the sea floor, and a storm comes episodically and smothers those organisms. And what we have then is the organisms, molds of the organisms are preserved on the base of these storm beds. So we flip over these beds and find fossils underneath. So what that means is that we have to flip over and excavate beds. So this is a really laborious process. We have been doing this now for 23 years, 24 years. This is not what paleontology usually is. Paleontology is usually go out in the field, tap on a rock, clink, 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 take it back to the museum. Excavating beds and flipping them over and looking at these snapshots of the seafloor, that is unique to Nilpena. It is unique to the fossil record. People do dinosaur digs and expose dinosaur bones. They're not in place, and they don't have bed after bed after bed of sort of the seafloor. So just to sort of give you an idea, this is a shot. We've excavated about 40 beds and counting. And you can see sort of in the lower left, we excavate the beds. They're various sizes depending on geology and whatnot. We then map the beds and log all the fossils and just literally get on our hands and knees and look to see what is there and what is preserved on this Ediacaran seafloor. And when we started, we thought we would be here for a couple of years. In fact, we had a grant that was 2 years here and then a year going to Namibia. That was 23 years ago. I haven't left. Obviously, I'm still here right now. Because of the nature of this record and because these beds were so different and so interesting, and every time we excavate a bed, we discover new things, sometimes new body plans, other times new ecological or behavioral relationships. So just to give you an idea of how long this is, my husband works in the Himalayas. So when our kids were little, they always came with me to South Australia. That is a picture year 2 with them in the wash tub in the shears quarters. And on the right, that's a couple of years ago in the Flinders Ranges. So this has been a very, very long time in terms of excavating these beds. So this research is based on many, many years of my work and that of my students and Jim's students and Diego students and so on. So what have we discovered? Sort of, hitting again that same theme, major ecological and biological breakthroughs, major sort of innovations in animal evolution we have in really sort of a 4-kilometer square area. But one of the really sort of to set the stage as if you close your eyes, maybe keep an eye on the slides, but as you think about, all right, what did the Ediacara seafloor look like? The first thing you have to imagine, if you're sort of floating around in the Ediacara Sea, is when you look to the floor of the ocean, it is not nice quartz sand like a sandy beach, but in fact is covered with sort of a ubiquitous, very organic, oozy sort of pond scum that was common in the Ediacaran because of the absence of organisms that moved and churned the sediment extensively. So if we go to the modern seafloor, we have things like the sea cucumber from the Great Barrier Reef, which is like a bulldozer species, and it's constantly eating and constantly pooping and eats all of the organics. Organics don't get a chance to accumulate on the seafloor. But if things aren't mixing the sediment and they aren't eating all the organics, you end up with something that looks like this. And while this is in the modern and this is the obviously Yellowstone, it's this kind of green ooze that in the absence of things eating. So that's your seafloor. And so this oozing scum, which some of these slides feels like it's sort of oozy and gross, you would not have wanted to put your foot into it. But this is the basis for the ecosystem. It's what things sat on. It's what some of them ate. It provided oxygen. And so this is sort of a very different paradigm than we have in the modern. So there's your seafloor. Where do we go from there? What do we have? What are some of our discoveries there? One of the first ones is evidence of mobility. We have at Nilpena the oldest evidence of something actively moving. And that is a really, really big deal in animal evolution. There are still things today that are sessile, that don't move, but think of all the many things that move. And think of what mobility does. We're all happy to be served food in a restaurant, but if that's how we always had to eat, we would starve. It's the ability to move and get food. It's the environment gets bad. You can move out of the environment. So many, you can find a mate. So the evidence of mobility is a really big deal. And this fossil here on the left, called Helminthoidicnites, is the oldest evidence of mobility on planet Earth. And it's a burrow. So it's not the organism, it's literally furrows, and they're really common that we find in on our sandstone beds. So we can look at that and we say, all right, in order to make that, it has to be something that's moving. It's a burrow, it's moving through the sediment. When we look at this particular burrow, the nature of where it moved and how it moved really requires that it's a bilaterian. So we are bilaterians. We are bilaterally symmetrical. We have a dorsal and ventral side. We have a head, we have an anus, we have a through gut, right? So we are bilaterians. Most animals, such as this dog, I think I put, yes, a lobster in here, are bilaterians, basically everything other than corals and sponges. So it's a big deal. So here we have evidence of mobility. It's made by a bilaterian, which is our closest relative. So here we then have our oldest bilaterian, and it is showing evidence of mobility. So for a couple of decades, we knew there was something that was making this burrow, but we didn't know what it was. And this is pretty small. You can see the scale. There are millimeter, 2 millimeters, and we just sort of was, yeah, it was generally accepted this was made by a bilaterian, but who did it was a mystery. Until we found the specimen in the lower left, where we actually found a little thing sort of the size of a rice grain near one of these furrows. And then with laser scanners, we're able to go out in the field and take the laser scanner out and scan, in this case, a bed out there covered with these little just divots. We're able to scan them and find this fossil here, which is we named Ediacara, which after Ediacara National Park, or Ediacara, which is meeting place, and Nilpena referring to the Nilpena pound. So this is a modular organism, a tiny, tiny little guy that was the first thing to move and is your closest relative, and was really, really tiny. As you can see, but a really significant fossil in terms of understanding animal evolution on planet Earth. And for something, you know, quite different, sort of the opposite extreme, is one of the ways in which organisms built skeletons in the Ediacaran was sort of tubular construction morphology. So think of a flexible straw, for example. And these guys were incredibly abundant. You may hear about lots of other Ediacaran fossils and think, oh, Dickinsonia must be the most abundant or whatever. But actually, these tubular forms were by far and away the most abundant, diverse sizes and types. This one in particular was first described as Form A, and it was first interpreted as fecal pellets. There's a lot of reasons, and if you look at some of these pictures, you might imagine why that may not be a good interpretation, such as the fact that they branch, or that they're all lined up in a row. So definitely not fecal pellets. So we did deep dives into this, going after this particular fossil, and realized that it was a tubular organism, which we named Phoenicia dorthea. This is, you can see the reconstruction in the lower left, and see both the fossils themselves and then their holdfasts, what it was that kept them on the seafloor. And when we looked a little bit closer and started looking at the holdfasts and the nature of these fossils, what we realized was that they showed synchronous aggregate growth, which is synchronous, single generation, aggregate, they are closely packed growth, they all grow together. So you might go to the seashore and see barnacles, same size clusters, or mollusks, mussels, same size clusters, where it's a single generation of organisms. Super, super common in modern tidal environments, on reefs and so on. Corals, very, very common with corals and other nigerians. So we felt we could, suggest that looking at this organism, that it might have two different ways of reproducing. One would be branching or budding, and then the other would be synchronous aggregate growth, which is the result of sexual reproduction in modern organisms. So for example, and you can, these are just a number of examples where corals release egg and sperm into the water column, triggered by the environment, triggered by lunar cycles, triggered by seasons, and so on. And this is a really good tactic because it reduces gamete wastage. It means that organisms can hold on to an area of the sea floor, and so very common in modern environments. So we published this paper, and this was Jim Gehling and I published this paper a while ago. And it was about the discovery of this organism, which is reconstructed here. And so there were lots of great articles, and then it just got, really picked up by the general regs. This is the Sun. I won't even read it, but you probably can. It was, picked up. This is the Sydney Morning Herald, tantrum sex in South Australia. So it got a lot of media play, but the reality is that, of course, sexual reproduction is a major evolutionary step in many organisms, including bacteria, but a big step in terms of animal evolution. And so we think a number of organisms were actually reproducing sexually, but in this case, we had really good evidence of sexual reproduction. And then this is a fossil we've discovered recently. This is a fossil that we had seen on a number of beds and called raisin, because it looks like a raisin. And often when we find a new fossil, we give it a nickname until we actually know it's a thing. And then we found a bed with about 100 of these. So then we realized, oops, we have to deal with this. And so this is, a number of these, and these are positives and negatives, and it gives you an idea sort of that it really does kind of look like a raisin. We then did a lot of analyses of it and scanned it, and then learned that these ridges that you see here are not ridges in terms of the morphology of the organism, but actually sort of deflation structures. And so we reconstructed it to this organism on the left, which we called Attenboroides jane. Attenboroides after David Attenborough, who has been here and done a lot for communicating first life to the general public. So this particular organism, Attenboroides, is the first animal multicellular metazoan in the water column that we know of. There are other ones that have been proposed, but they keep being put back on the benthic sea floor with new evidence. So right now, this is the only animal that we know of in the water column. So one of the things that when I first started, I wondered, would I have needed fins in the Ediacaran? So you go to the Great Barrier Reef or any place along the coast and you wear fins because you want to go look, oh, look over there, there's something else and so on. So would you have needed fins in the Ediacaran? So the short answer is you definitely would have needed fins. When we started off, I thought, oh, we're going to excavate 4 or 5 beds. They're all going to be much of a muchness, done and dusted. Let's go to another continent. We're still excavating beds. They're all different. Same characters, but in different body sizes and different population structures and so on. So we started to look at how patchy the seafloor is. And I know since this is a geography group, I thought I would put in some marine geography in terms of thinking about how patchy the seafloor was. So looking at heterogeneity of the seafloor. And we think about this with respect to forests, with respect tropical rainforests, with respect to reefs and so on. But because we have these snapshots of the Ediacaran seafloor, we can actually think about it with respect to the Ediacaran. So this is a figure that I actually put an old one in. The new one is way too wide and too complicated. These are two different environments above the top. Each one of these bars is a different bed. So this is a number of our beds. And all I want you to take away from here is that there are a bunch of different colors, and that a lot of these beds have very different colors than other beds. Showing, sort of unexpected that they have different abundance and diversity structures, sometimes stacked on top of each other within very similar environments. And just to, for those of you who might think about things like this, and I will explain it, is thinking about how does the patchiness of the seafloor in the Ediacaran relate to the modern. So one of the things we think about when we think about modern diversity is what we call alpha diversity. What's the diversity of this community within this rainforest? What's the diversity of this single reef part of the Great Barrier Reef? So that's we would think of as alpha diversity. And then beta diversity is, okay, let's take this part of the tropical rainforest and look at this part of another part of the tropical rainforest. And how do they compare? What's the combined diversity between them? And that's a way of thinking about beta diversity. And that's a way of capturing patchiness. If your reef varies from one side to the other completely, that would be high beta diversity. If part of the tropical rainforest is really, really different from another part of the tropical rainforest, that would be high beta diversity, high patchiness. So what you see here is pretty low, and don't worry about the numbers on the axes, but quite low alpha diversity compared to modern samples. And these are dredge samples collected from all over the world with all sorts of error bars and everything else. But I just sort of show you this in terms of thinking about space, is that Nilpena and two boxes or two different environments at Nilpena have extremely high beta diversity or extremely high patchiness with respect to both the rest of the fossil record, as well as the modern. And so these are some of the kinds of questions that we're asking and can sort of go after and think about as we have these crazy beds that we are actually able to excavate and analyze and think about the evolution of life, and not just new body plans, but their spatial orientations with each other. So just to sort of finish up, some of you may know this, I think we have some from South Australia along with this, that Nilpena Ediacara National Park is part of a Flinders Ranges National Heritage Bid. We are on target to be the 2025 Australian submission for this category. And it doesn't just include Nilpena Ediacara National Park, but fossils both above and below. And so again, going back to thinking about how does life evolve on planet Earth, here in the Flinders Ranges larger area, we have an incredible record of the incredible environmental swings that were happening, snowball Earth and warming and cooling, leading up to the early record of animal evolution, the Ediacara Biota, and then the Cambrian early record of Cambrian life after that. And so just stay tuned. And hopefully we get this World Heritage. And so when you come visit the park, you can come visit other World Heritage sites. And just this is the sort of Nilpena part of that, is that Nilpena Ediacara National Park represents, this unprecedented excavation of these beds, which allow us to do really look at ecological scale patterns that are usually unavailable in paleontology, that are reserved for the modern, like looking at how organisms live relative to other organisms and population structures and whatnot. So I will leave you, the way I started you was, the way I started was, this is a time lapse taken about 10 meters from where I am. Thinking about life on other planets sometimes, you know, to pause and say, what an incredible record that we have on planet Earth of the unfolding of life on planet Earth. And we have an incredible fossil record, including many in Queensland, and Nilpena represents just one little tiny time slice that gives us insight into this early evolution of animal life. And with that, thank you very much. Just some pictures of the many people who have helped over the years. Thank you.
Okay. Well, thank you so much, Mary. I don't know whether John wants to invite us to engage in a conversation. John, do you want me to lead this part?
Go for it, Diego, all yours.
Okay. Well, thank you, Mary, for that presentation. It's very, very illustrative. I think we've all learned a lot. I think it's been a combination of biology and geology, and I think that it's been made very accessible to our audience. While I was listening to you, but also from my previous experience and as a Cambrian and now an Ediacaran worker, I've come up with a few questions. Are you okay if I present those to you?
Absolutely.
Okay. So you mentioned World Heritage. I was doing a bit of research for along these lines, and just to share with our audience, there are about 1,200 UNESCO World Heritage sites in the planet. Out of all of those, there's only 16 that are based on fossils. So what we're doing here with Nilpena Ediacaran National Park and Bratchina Gorge and some of the other sites of this, we are doing something very important. The other ones that relate to this are the Burgess Shale fossil site from Canada in the Cambrian, 1981, and then Shenyang in China from 2012. There's only one Ediacaran one in the planet, and I'm sure some of the audience will know which one that is, Mistaken Point, in Newfoundland, from just 2016. I wanted to ask you, this is imminent, but what do you think it's going to bring the amazing level or the stamp of approval of UNESCO to what South Australia has to offer in the Flinders Ranges? What do you think it's going to bring from a social, economic perspective, as well as we can go into the cultural as well, obviously?
Yeah, I mean, I think this is, well, in part because it is a 7-site, so it's a serial site World Heritage bid. So it includes Arkarula, which many of you may have heard of, and a number of sites, Nilpena, and then a number of sites in the Flinders Ranges. A lot of these sites are easily accessible. Number of them are free. And so I think, you know, people are interested in visiting World Heritage sites. And this is an area where the coal mine was closed a number of years ago. There are great facilities up here in the sort of greater Flinders Ranges area. It is an absolutely gorgeous area, even nothing to do with the fossils. So I think that it will bring a lot of people to the area. International travelers coming to Australia hopefully will find themselves in South Australia. And I think that's the hope here. And we're starting to see the impact of Nilpena Ediacaran National Park already on tourism in the area. So I think World Heritage will only help. And obviously, it's great for the conservation and preservation of many of these sites, including Nilpena.
My experience is that from the World Heritage sites that I've been, and I've been to Burgess, Shenyang, and Mistaken Point, is that they cross-pollinate. A lot of, as you say, people collect, quote unquote, visiting UNESCO World Heritage sites, because things like the Great Barrier Reef or Uluru or Notre Dame in Paris, or the Sydney Opera House, those things are so iconic that people want to go there. Is there anything in the plan to sort of present when this happens and share some of what we have here with other UNESCO sites, so that when other, say in the US or Canada, when people visit those, they say, oh, we really got to get to Australia, because that's going to complete the picture of our understanding of early animal evolution.
Yeah, I mean, there is long term, of course, we have to get it first. So
Absolutely, but I think this is not so much.
There is an idea to do that. We don't want to
step ahead of ourselves.
Yeah, we don't want to do that. You don't want to start saying, oh gosh, when we get it, we can do this because we don't have it yet. I think we are an obvious link between Mistaken Point and the Burgess Shale. Mm-hmm. And that's, you know, and there are, the Chengjing is also part of that, but those three are, you know, readily accessible in a way. And I think connecting this to other sites in Australia is there's already thinking about that. The
Naracoorte, for instance.
Naracoorte, yes, and I think an even Great Barrier Reef as we look to ancient and modern, yeah. And so, and the person who is ultimately pulling together our words and making them readable for UNESCO did the Sydney Harbour Bridge, not the Sydney Harbour Bridge, the Sydney Opera House bid, and a number of others in Australia. And I think he's very cognizant of the need and also fun of tying it into other World Heritage sites. But we definitely aren't working on that yet, but we will when it hopefully happens.
Great. I think that I saw a symbol of NASA in one of your images, and we've got a NASA project. Do you want to share with the audience how you mentioned One World, and what is it? You've got a project on searching or using the Ediacarans for looking at what we might expect in other planets in terms of, you know, early life. Could you elaborate?
Yeah, so I think, you know, most people think it's really strange that a paleontologist is funded by NASA. I'm currently the only one. But that's because we've run the experiment of the advent of animal life on planet Earth and how that unfolds. And so I've had NASA funding for about 20 years, and we've asked a lot of different questions, like, you know, do the little guys like Icaria and scanning and doing things like that. Right now, we have a grant that Diego's part of, where we're actually looking at the nature of how sediments are packaged. So when you look at sedimentary rocks, sandstones and whatnot, how they are packaged in the Ediacaran because of the presence of microbial mats versus other times in the geologic record. And it turns out microbial mats have a very strong signature that you can see from the side, not just from above looking down. And so we work with some extremely clever engineers who have developed machine learning or AI to recognize these structures from pictures. So we've used pictures from Nilpena to train the AI, and now we're looking elsewhere to see how, you know, to keep training and testing it. And, you know, we're actually sort of fiddling with pictures from the Curiosity rover on Mars, and no, there isn't microbial life on Mars, but just how it might work on a rover. So it seems pretty far-fetched, you know, to be saying, oh yeah, Dickinsonia is relevant, but how an animal moves, how you have a muscles, what does the oxygen levels need to be for animals and so on is very relevant as we look very far ahead, though I don't think, pretty sure we're not going to find a Dickinsonia or any of the Ediacara Biota on another planet, but thinking about how animals evolve is important when we think about things like that.
One of the things I noticed is how a lot of the new genus, genera, and species that are described have names of important people, people that are related to the research or the outreach of some of the findings. What our audience might not know, that in general, there's three ways of naming this. You never name a species or a genus after yourself. That's not acceptable. but basically you name it after somebody, you name it after somewhere, or you name it after a feature in the organism that is a diagnostic that allows you to recognize this as different from something else. You mentioned Attemborietes. Are there any other species that you've named after famous people? As you know, I think that some people might be interested in hearing that.
Yes, well, one of our species is called Obamas, after Obama, and that was in sort of 2016. And it was a if you are familiar with Barack Obama, he's known for sort of having big ears. And it is a fossil that actually has a torus shape or is not unlike an ear. So we got off on that track. And then also because of his contributions to science. It's the only sort of famous American we've named after things. We do a lot of local localities, use Etna names, or name, Diego is part of a new species of a fossil that's named after our colleague, Jim Gehling. But Obamas, we just couldn't resist at the time, and also because of the nature of those fossils. So sometimes you can have a little fun with them. Phoenicia is Latin for rope, and that fossil, this first sexual reproduction sort of had its nickname was Ropey. And so that was sort of natural. And then Dorothea was actually after my mom because she came when my kids were little for years and years and years and took care of them while I was off doing field work. So we have various reasons for naming them after people.
Yeah.
Places.
What did Obama actually comment on the fact that you named a fossil in the shape of an ear after him? Did you ever
never said, and in the fossil description, so I know this is being saved to YouTube, but it's long. We do not say that it's because of his ear. When we wrote him a letter, we said because of your contributions to science. So the ear thing appears nowhere.
Right. But he's a smart man.
But this is now going into the Obama Library in Chicago. And so we've sent them the, Australian Journal of Earth Sciences and replicas and so on and so forth, and the letter that we sent to Obama. So it's become a whole thing, which is only fun. Yeah. And he really did contribute a huge amount to science and science education and science appreciation in the US. And of course, we're funded in the US, so.
That's absolutely very fitting. You mentioned species. From the area of Nilpena that I work on, there are new things. I'm not going to say appearing every year, but there are new things coming out. What's your estimation of number of species that you might have found, but you don't have enough specimens to actually name them yet? So what can we expect in that area? How many new species do you think you've got, besides what you might find in the future, which we don't know yet? But how many do you have that still need to be named?
We have a few that, as you know, are in the publishing phase. So we have a couple there. We have a few that we're fiddling with, probably 3 or 4 that we're sort of like, is this or isn't this a thing? There are times when I think that we are so underestimating the diversity here, because we, tend to lump things. On the other hand, I think between all of us, we probably get it right. We still are finding really different body plans. So, you and I are part of a paper that is totally wacko body plan, where it's an organism that literally has a question mark on its dorsal side. So, there are some very weird things, which makes it fun, and things like Acaria that we can firmly put with animal life. So that's very helpful.
How do you see future research in Nilpena in particular, but in the Flinders Ranges in general, in say 5 or 10 years? What do you think is going to happen?
I think we're going to continue to find new things. So there's a level of discovery. I think we are really getting a handle on where these organisms lived and why they were preserved, and that's important. I think overall, we're going to get a better idea of how they fit into animal evolution. I think tying them to the global environment is important. Oxygen levels, which are something we can get from other places, but not as well from here, is important. So I think tying this area into the global record will be, as we get better at correlating, I think will be really important. There's, the Flinders Ranges has a phenomenal succession through this whole interval, and I think tapping into that to set the stage before and after the Ediacara Biota will be really important.
You in one of the papers, particularly the one on Icaria, you used micro scanners. How do you think that technology is basically and also computational tools, AI and others? How do you think what sort of role do you think that is going to play moving forward?
Yeah, so I think things like laser scanners, particularly this is a medical scanner. It has no business being in the field. We have all sorts of ways of powering it, as Diego knows, because he uses it as well. That's been a major breakthrough for us, for all of us working here in terms of understanding the animals. So I think that's really important. Computationally, we're able to go after spatial issues that we didn't used to before. So understanding, so you might go in modern environments, think about whether organisms are living aggregated or clustered, whether they're segregated, whether they're distributed randomly. We have huge amounts of data for this, and I think that also is something that we're able to do both in terms of not just the technology, but the development of statistics in ecology that we can adapt. And I think that's been, those kinds of things have really changed the way that we think about this. We really are asking ecological questions of the fossil record. And so that's pretty amazing. And so it's keeping the new techniques that are coming out in ecology that are sort of changing the way that we do a lot of things here.
Well, one of the things that our student Tori Botha has began to dabble in is use of geometric morphometric, which is defining structures, features in an organism to try to understand how they grow, how they are affected by the environment. These are, as you mentioned before, 100% soft-bodied organisms. There's no shells as such. There might be some very fine spines in the case of some of the organisms. They might, as we get into the youngest or Ediacaran, there are proper tubes, etc. Do you see that is basically the way that we also want to start thinking level, utilizing some of the fossils we already have and taking them to the next level with technology?
Yeah, for sure. I mean, I think doing the morphometrics and geometric morphometrics is those kinds of things. Yes, again, taking it from the modern, or even fossils that have skeletons, to figure out how organisms grew, how they lived, and really looking at these fossils as living organisms rather than just weird things that lived a long time ago, I think is really changing the way that we can address these organisms and think about how they lived.
Okay, well, I'm happy to, if you're okay with that, there are a few questions from our audience. Ralph, for instance, says, how does the Ediacara Biota of South Australia compare with the Ediacarans from Newfoundland?
Yeah, so the Newfoundland is older, so the mistaken point fauna is older. So the sort of generality of what's found in mistaken point, which is the oldest evidence of animals and the earliest record of the Ediacara Biota, those organisms are what we call fractal. But really what they are is, if you think of how your lungs are, that your lungs maximize surface area for gas exchange. So we have a whole suite of organisms that are built like that in Newfoundland. And then they dominate for a while, and then comes in this sort of, as we call it, the second wave of the Ediacara Biota, which includes Nilpena and the White Sea of Russia, the Ediacara Biota of the White Sea of Russia. And we think that the White Sea and Ediacara member in South Australia are about the same age. And so while those same fractal organisms, lung-like organisms, continue through and we find them here. We just have a much greater diversity. That is an incredible record and worthy of World Heritage, which it is now. So that's great.
Another question, this time, Stuart. You mentioned the rock is sandstone. Is there any finer sediment that allows cellular scale preservation and comparison with present cell structures?
That would be my dream. That certainly is not the case here. Grain size is such a limiting factor for us. So no, we are sand, and sand. So while in my mind we have a perfect record, it is imperfect. There is certainly in other places where you get shale, such as in China, the Burgess Shale, literally a shale, where you get finer structures preserved like this. Cellular scale is really a very distinct type of preservation. You can get phosphatized cells and things like that. We don't get any of that here, so we don't see any of the cellular structure here, sorry to say. But you can elsewhere in the fossil record, in both older and younger rocks, of individual sort of eukaryotic cells.
Kay was asking, does the biota of the currently known Ediacaran sites support that these would have been much closer, I would imagine, geographically together during the Ediacaran period?
Yeah, Kay, great question. So yes. So during the Ediacaran, Australia was up by the equator. The White Sea was, or that area of Russia was definitely closer to where I am sitting right now than it is today. These organisms still, we get the same species here and in the White Sea of Russia, which is crazy. So they were certainly much closer. It also tells us about the dispersal methods of some of our taxa, whether it's the similar, because even though they were closer, you're still crossing a little bit of an ocean. They weren't attached, but they were definitely much closer. So yes, great question.
Kay also was mentioning, as we get closer to the Cambrian period, was there a specific event, or was this a reasonable expectation of the evolution of life between the Ediacarans and you know, the Phanerozoic faunas that we're familiar with? Apparently, there has been some fairly savage extinction events during the Ediacaran period. So, what was different between the Ediacaran and the Cambrian?
Okay, so that's many people's careers, including the two of us who are on this Zoom. So I think most paleontologists and biologists would say there are no expectations to the evolution of life, though others might disagree. What we see is this is the first experimentation in animal life on planet Earth. You know, you have the mistaken point, and you come to the White Sea assemblage, and you see what we have here. Standing in an Ediacaran sea at Nilpena, you wouldn't necessarily have predicted the outcome and that you would end up with the Cambrian. We do have whole body plans that go extinct within the Ediacaran, like triradial symmetry. So I think animal life is evolving on a backdrop of a really dynamic planet where we know oxygen levels are changing pretty rapidly. So that what we think is going on just after the White Sea, so nice diverse time of the White Sea and the Ediacaran member, and then oxygen dropped. And a lot of things that were around at Nilpena go extinct, and that's just environmental change and drop in oxygen. And then we have things in younger elements of the Ediacara Biota where we get a few more mobile organisms. Oxygen is still low, but sort of increasing. We have more fluctuations in oxygen. And then move into the Cambrian, building on some of the body plans that are established in the Ediacaran, both during the time of Nilpena, but also in the time that is just younger than this. that's represented in Namibia. So what's different? Probably oxygen is coming and going. What's different is in the Ediacaran, your experimentation is on really fundamental body plans like Cnidaria versus Porifera versus Bilaterians. In the Cambrian, you're getting a lot of experimentation within clades, within the arthropods, for example. And while there were oxygen issues in the Cambrian, it was nothing like you were getting in the Ediacaran. And so once things got started, even though we had a lot of extinctions, we didn't lose really whole body plans again. The trilobites go extinct, but arthropods are still around and so on. I don't know if that answers your question, but we worried these questions keep us up at night.
Ray was asking, are there any updates on whether our Karua is the precursor of vertebrates? I think our Karua, I don't think Karua has been recognized or suggested to be a precursor of vertebrates. I think that he might be mistaking our Karua with that protochordate that Jenkins discovered, which has been illustrated in places. Our Karua is considered with its pentaradial symmetry to have been related, if anything, to echinoderms, so sea urchins and sea stars and their relatives. But so if Ray wants to elaborate on that, that'd be good. John was saying from the photos of the site, it seems to be on a similar level in quite small area. Given your discussion of the diversity, does it represent a long period of time or a wide range of environments? Or is the site much, much larger? How big is the study area that we're working on?
Great question. So the Ediacara Biota in South Australia covers hundreds of kilometers. Our particular site is pretty small. It's about 6 square kilometers. And it is, and you saw a site of a hillside, which has many of our beds. So while the fossils occur throughout the Flinders Ranges, Nilpena has a sort of distinct and very helpful geology, which allows us to do this excavation, which is not possible elsewhere. So the fossils occur elsewhere, but the ability to excavate beds right now is really in these Ediacara hills that cover a larger area, but where you get these fossils occurring is a much smaller area. It probably doesn't represent that much time at all. What we do have a number of environments, 4 or 5 sort of, we sort of from very shallow water to much deeper water. And so it is a remarkable diversity in this relatively small area, which is why it's even more surprising, because we have, these 40 beds, which, are within really a 2 to 3 meter interval of rock. And probably, maybe it's 1 million years, not evolutionary time, but rather ecological time scale. So yeah, it's a crazy amount of diversity, which is why we're still here. 20 years later, still excavating beds and still discovering new body types.
One of the questions that we get when we show Nilpena, when we guide it, is how long has it taken for those beds to deposit? And something that is counterintuitive is the fact that, for instance, comparing Ediacara with, say, the Emu Bay Shale. In Emu Bay Shale, you might have a unit that's this thick that has been one single storm event of 2 days, and you might have a bed that's this thin that might have taken 3 months to deposit. Do you have any information, any resolution to tell time in between your beds? I mean, obviously the older ones at the bottom and the youngest at the top. But do you have any way of telling time, maybe with the maturity of the microbial mats?
Yeah, so one of the things that is remarkable about this record is that we can even ask those questions. So we look at it, we have a series of beds and we look at the nature of the beds, but we can also look at the nature of the communities that are preserved. So if you have a really mature microbial mat, if you have a population structure that has clearly a number of generations of taxa, if you have high evenness, which is, you know, the bed isn't dominated by one thing, but it's, you know, quite mature community. Those are the kinds of things that give us an idea of the amount of time, relative time between depositional events, which is amazing to be able to think about things like that. This was a protected bay. We don't think we had huge storms coming in all the time, but lots of them were very little and would deposit a millimeter or two of sand, and these organisms would probably just shake it off and live through that event quite easily. So we do think in terms of relative time within the succession, and it doesn't represent a huge amount of time. It's quite a window.
Yep. One of the things that Mary and I are working on, we've got a project from the Australian Research Council to try and date the Ediacaran fossils from the Flinders Ranges. We know that the Russian fossils with the same species, Tribrachidium and Andromeda, or et cetera, some of these things, Yogia, Dickinsonia, some of these bits are dated as 555 in Russia. What would you expect to find for us here, taking into account that, you know, yes, some of these taxa were actually surviving for a long time?
Yeah, no, I think, you know, species today average about 2.5 million years in duration. We correlate with the White Sea because we imagine that we have the same species. So it is, give or take, 2.5 million years. We have the same suite of species, and it's how geologists correlate rocks throughout time. So I think that we're on the right track of relating this and assuming that we are of similar time with the White Sea and Russia. None of us will be surprised if we're a little bit older or a little bit younger, or between, you know, or above and below or whatever. I mean, I think these are questions that we're hoping to answer. And I think we will get better clarity on that. And going back to your question of research in the future, I think being able to get a handle on the age would be fabulous. And I think we'll work towards that.
Okay. Okay. Can you explain the significance of placing the golden?
It's a golden spike.
Yeah, spike of okay. Yeah, the significance of the golden spike for the Ediacaran in the Flinders Ranges.
Yeah, so for those of you who don't know, and actually if I can share my screen again. I actually have a slide on the golden spike. So the way that we do, that we think about geologic time, you have to, you know, when we think about our day-to-day life, it's divided into hours and days and weeks and so on. And that's how we both communicate as well as sort of structure our lives. And in order for geologists to communicate, for us to talk to people in Kazakhstan about what age rocks we have divided geologic time, particularly the last sort of 635 million years, into what we call periods. And there are 13 geologic periods. And largely, they are designated by major turnovers in the history of life. So the end of the Cretaceous period corresponds to the extinction of the dinosaurs. It's a major biological event that we can recognize. And each one of these period changes, boundaries, has a place in the world where there is the exemplar of that boundary. And so The first period to be described in the last literally 100 years, human years, was defined as the Ediacaran period. So this is the last bit of the Precambrian and before the Cambrian. There's only 13 boundaries. It's the only one in the Southern Hemisphere. There are none in the United States, if you're wondering. So these are picked for places, and there's huge committees, and it's highly political, and there's so much arguing, and everybody goes nuts trying to get it because everybody wants one of these. And so the base of the Ediacaran period is defined in Brachina Gorge, which is not very far from where I am, in the Flinders Ranges. And it literally, you can see this, it's not really a spike, but it's called a spike that's been driven into the rocks that represents the base of the Ediacaran period, and it's defined by the end of a huge global glaciation and the beginning of a warming that happened after that. So normally they're defined by biological events, but there aren't those kinds of biological events. So this is a really big deal.
One of the things that people don't normally realize, I don't know if you could get back to that slide, is the fact that this is 635 million years ago. And our Ediacaran fossils are 555. So there's a very, very long time in the Ediacaran, most of the Ediacaran, where there are no macroscopic fossils. So the Ediacara Biota is only the last, say, 30 million years of the Ediacaran out of the whole extent of that geological period. And that's something that people, I would invite people to visit Brachina Gorge, where you can see the GSSP, you can see the Ediacarans, you can see the first animals in the form of the reefs made by archaeocyaths, which are marine sponges. And so it is, and you can see the glaciation, stromatolites, you can see the whole change. I see it, I would imagine that Mary also has that feeling that Brachina Gorge is like the backbone of the nomination, and you can look at details of that backbone of that time sequence in Nilpena Ediacaran National Park, where the fossil beds are almost horizontal, and that's what allows us to excavate them. Because if you see it in Brachina Gorge, they're almost vertical with rocks on both sides, so you can't excavate. Right, so that's why for the Ediacarans you go to Nilpena Ediacaran National Park. For the archaeocyaths that we've got other sites, Ajax Mine, etc. So it is, Brachina Gorge is definitely something worth visiting. And if you get to visit Nilpena Ediacaran National Park now, it can be visited both through DEW, Department of Environment and Water, but also through the Prairie Hotel in Parachilna. So there's a lot of opportunities to get engaged more and more with what's happening there. Arkarula is open to the public as well. So these are things that people should begin to consider if they're thinking of coming to South Australia. Come and visit, the Flinders. So there was one last question that I could read. How thick are the strata that we are finding useful? And how many useful levels do you typically find?
The beds we're excavating are on the order of a centimeter to about 5 centimeters. We have ones that are thinner. We definitely have ones that are a lot thicker. So the useful ones are the ones that are easy to excavate. And we do it all by hand. We don't break rocks to do this. But they, we have one of our pits on the hillside. We probably have 8 really good beds and a handful of other ones that we've excavated within that succession. So with any given sort of succession, that's probably about how many we would get, 8 to 12 beds, in an excavation pit.
Okay, well, thank you, Mary. John, I think we've gone over time compared to what the original running sheet was, but I think we've had a fantastic conversation. There've been a lot of really interesting questions from the audience. Thank you very much for sending those over. John, I want to thank Mary, and I want to thank you for inviting me to participate in this, John. I think that if you consider coming to Adelaide, definitely visit the museum as well as the Flinders Ranges.
Mm-hmm.
Don't-
Very much. Diego would like to second that. Thank you to both yourself and Mary. Fascinating conversation. I think we've all learned a lot. So for everyone online, please join me in thanking both Diego and Mary. And with that, we will wrap up for this evening's webinar.
Great. Thanks very much for having me.
Thank
Thanks,
you.
everyone. Bye.
Bye.
See ya. Bye.